Hello, Dr. Yu. Very nice meeting you. So we're interviewing with you because your abstract was selected as one of the top-rated abstract in this BCVS 2020. Dr.
Yes.
Yu, could you introduce first yourself?
Hello, I'm Ming Yu from the National Taiwan University, the Department of the Pharmacology. And I just graduated from the master degree. Yes.
Could you briefly summarize what your findings are?
Yeah, so our study is that we identified a novel cardiac long non-coding RNA that's called Lnc-SYNPO that is upregulated in response to the pathological stimulation, such as the high concentration of endothelin 1 and the myocardial infarction. So we use the human-induced pluripotent stem cell that's called iPSC-derived cardiomyocyte to show that Lnc-SYNPO function as a negative regulator of SERCA2a, where Lnc-SYNPO repress the expression of SERCA2a through recruiting the polycomb repressive complex that's called PRC2 complex, which is the epigenetic modifier. So it can lead into the epigenetic silencing of the SERCA2a gene. So this was the first study to uncover the epigenetic regulation of the SERCA2a by a cardiac link RNA in the failing heart. Also, our finding offers a new strategy to maintain SERCA expression in heart failure by targeting Lnc-SYNPO. Yes, that's our.
That sounds great. So people have used kind of a direct SERCA2 gene therapy and show some favorable results. And so I think your findings are really impressive. But we just want to know how you came up with this idea. How did you discover this long non-coding RNA?
Oh, that's a very nice question. Because there is a lot of current evidence supported that link RNA are involved in the cardiac development and the progression of the cardiac diseases. So we would like to discover the link RNA such like that. So we perform an RNA sequencing experiment on the human cardiac tissue from the failing patient and the non-failing patient, and then conducted a co-expression network analysis to identify link RNA that could possibly be involved in the regulation of the calcium homeostasis protein, such as SERCA2a. And then we perform a series of in vitro and in vivo experiments and confirm that Lnc-SYNPO actually participate and involved in the regulation of cardiac SERCA2a gene and calcium homeostasis. So that's
okay. That's terrific. So so you must have through this a comparative analysis, you must have found a lot of other targets. But how did you come up specifically, focusing on this specific link RNA? Were you interested in, from the beginning, finding some, upregulators of this SERCA2a, or, I mean, upstream regulators of SERCA2a, or not?
In the beginning, there's a lot of link RNA are correlated with SERCA2a. There are some positive correlated and there are some negative correlated. But after some functional testing, like using a lot of shRNA to knock down the link RNA in the cell, we choose the Lnc-SYNPO because it is the more promising and the more effective one.
I see. So you've done a lot of screening experiments and then ultimately found this specific molecule, right?
Yes. Yeah.
Okay. Great. So you mentioned that this may be a good kind of option for future treatment of the heart failure. Do you have any specific sort of plan, or do you have any idea how you can apply this for the treatment of heart failure in the long run?
The importance of this study is that the study is the first to report that the first link RNA that are involved in the epigenetic silencing of SERCA2a gene and regulate the calcium homeostasis in cardiomyocytes. So this result will provide us a better understanding of the complex regulatory network involved in the calcium homeostasis and the calcium handling in the cardiomyocyte. And the important is that because we can easily utilize the antisense oligonucleotide, which can directly target link RNA in the cardiomyocyte. And so we think it will become a novel therapeutic approach that is completely different from current heart failure therapeutics. And this will also replace the current approach because using the viral vector to boost the SERCA expression in heart failure patient is not effective and maybe there are some defects. So we think that using the link RNA as a target may be a good choice.
So yeah, I think I'm going to just briefly summarize what your findings are. Dr. Yu's group actually compared the gene expression arrays between the failing and non-failing hearts and identified this non-coding RNA Lnc-SYNPO, which is transcribed from the 3' untranslated region of SYMPO2 as a potential regulator of SERCA2a. This novel link RNA controls SERCA2a through binding to the EZH2 and SUS12, the two core components of the PRC2 complex. And this actually controls this histone methylation and then controls SERCA2a. Thank you for your outstanding work and congratulations. You're beautiful. And also I'd like to congratulate on your mentor as well.
Thank you, Professor Yoon.
Okay, keep safe and well during this COVID challenges.
Thank you.